now that you know all about the macromolecules and you're familiar with the monomers and the polymers, this lecture is going to focus on how we actually take the monomers and make them into a polymer, and then later how we can take that polymer and break it back down into the monomers again. Okay, so that's what we're doing with this one. So in order to make a polymer, you got to take those individual monomers, those individual building blocks, and you have to bond them together. The name for this type of reaction that bonds two monomers together is called dehydration synthesis, okay? And some features of dehydration synthesis is that we would call this reaction an endergonic reaction, which means it requires an energy input, okay, because we're forming bonds here. And this is also called an anabolic reaction. And you might have heard of anabolic before with like anabolic steroids, and you think of that as like someone who's like really jacked, right? And so the way that I remember is like anabolic making bigger. So we're making a bigger molecule, and or anabolic, atabolic, however you can remember that, okay? And just as, just to keep this in the front of our head, the bonds that we're making here are covalent bonds, okay? So here's a little drawing of how this works. It doesn't matter which macromolecule you're talking about. It works the same for all of them, okay? So let's just, this is some kind of macromolecule, okay? And here's another one, and we want to make it longer, let's say. And so notice that, and this is true for any of your macromolecules, they all have H somewhere and an OH somewhere else. So the H and the OH are going to come together, and this is going to form a water molecule. When we take this H and this OH, now these spots are available, and now these two things can bond together. So this 3 is going to be with this one, and now here it is. Okay? So notice that the orientation is really important because you have to have H and OH next to each other so they can form that water molecule. Okay? Because if they weren't there, then they couldn't form the water molecule, okay? And then, so here's why it's called dehydration synthesis. Dehydration, because essentially we're removing a water molecule from our monomers, and synthesis, because we're making something new, we're building something, okay? So the name kind of tells you what's going on. Here's an example with an actual monomer instead of just like a generalized diagram, okay? So this is like conceptually what it is, but here's what it looks like with proteins. So here's an amino acid with an R group. Here's another amino acid, but the R group could be going up or down, doesn't matter, okay? So this H and this OH are going to form a water molecule, bloop, and it's going to go away, and now where they were, now these two things are going to be bonded together. So the carbon and the nitrogen are going to be bonded together. And notice down here, we see that covalent bond in green. Okay, and here it is again with this H and this OH, so now this carbon and this nitrogen are going to be bonded together, and so that's shown in green. Again, covalent bond, and here's the last one. There's the bond in green, and then of course this would produce 3 water molecules, one from here, one from here, and one from here. Okay, so that's how we could take 3 amino acids and link them together. So why would we be doing this? Like, why would our bodies be doing this? Well, there are a couple reasons. If we're going to make a new cell, we need to build all the things. And so we're going to need to be making a lot of big molecules, okay? If your protein is needed, like a specific job is coming up for the cell, it might need to build this protein for this job. And so if it's going to need to build a protein, it's going to need to do dehydration synthesis. And also if you're storing energy, if you got some extra energy, you want to store it, you're going to create that lipid or a more complex carbohydrate. And so you would link some monomers together for that reason. So now that we know why you would create a polymer, let's think about why you might break down a polymer. So let's keep these reasons in mind here. So for breaking down a polymer, maybe it's time to use that energy up, that energy storage, that fat storage. Well, now we need it. We skipped a meal or what have you, we're going to need that energy, and so, bam, we're going to break down the polymer. Maybe it was made wrong, and so we want to be able to break it down and start over again. Or maybe it's old. Maybe the cell's old and it's dying, and so we're going to get rid of it, so we break stuff down. Or if that protein that we made, it's done its job, we don't need it anymore, your body's going to be really efficient, and it's going to break it down and use those pieces to do something else, okay? So that's why we would break them down. And so now the process of breaking down a polymer is a different reaction, okay? This is called hydrolysis. When we take a polymer and we break it down into a smaller polymer, or we break it all the way down into individual monomers, okay? By definition, the way we could describe hydrolysis is that it's an exergonic reaction. So we release energy. So notice this is the opposite. Whereas before it required, dehydration synthesis required an energy input, and that was called endergonic. This one's called exergonic. Yeah, lots of vocab, okay? Before, it was an anabolic reaction. We were building things bigger, anabolic, atabolic, right? Well, now catabolic, we're breaking things down into smaller pieces. The way that I remember that catabolic is this one is that catabolic, catabolic. So I'm snipping things up, okay? It's just a good mental image. And so this is how we would describe hydrolysis. So let's see what that actually looks like here. Okay, so we're breaking these covalent bonds. So here's a general picture, right? Here's whatever monomers of whatever macromolecule we're talking about. And then we add water in, okay? Hydro is a prefix to mean water. Lysis is like to break. So literally we're taking water and we're busting something open. We're breaking something apart. And the thing that we're breaking is a bond. Okay, so in this case, we're breaking the bond between 3 and 4 with a water molecule, and an H goes on one and an OH goes on the other. Okay, now you don't really need to worry about this, but the H goes on this one because the OH is dangling off this other end, and the OH goes on this one because the H is dangling off of this end. There's always going to be one with like those different dangles. And yes, dangle is a technical term. Why? Because I said it was. So that's hydrolysis, the exact opposite of dehydration synthesis. Instead of removing water, we are adding water in to break those covalent bonds. All right. So if you want to check this out in the textbook, which I always recommend that you do, this is chapter 3, section 2. And then just some good review questions for later. If you can answer these and do these things, then you understand this topic and you are good to go. If not, make sure to write down your questions and we'll make sure to get them answered in class.